We're now moving on to our special speakers, and our opening address today is, I'm very excited to say, is from Professor Steve Hopper. We'll have time for questions at the end, so Steve can talk to us about his reflections on threatened plant conservation in Australia since the 1970s. And this is such a timely talk for us, Stephen, because and I hope you can write something about this for us, for our journal even, because I don't think anyone's really pulled this together. Sorry, setting big boots. But Professor Hopper served as Director of the Royal Botanic Gardens, Kew, and as a Director of Kings Park and the Botanic Garden in Perth. He's been involved in flora conservation research and academic teaching in conservation biology at the University of Western Australia, where he currently works as Professor of Biodiversity at the Albany campus. That sounds like a bit of a lurk there, Steve. Professor Hopper investigates the ecological, evolutionary, conservation and sustainability aspects of biodiversity on old, climatically buffered, infertile landscapes. Bills, I've learnt today. He has a special focus on plants on granite rock outcrops and on the Hemodoraceae, eucalypts and orchids, and he also leads a small team researching links between Aboriginal and scientific knowledge systems of biodiversity with the Noongar Aboriginal elders. Could you please join me by welcoming Steve for his presentation?
Thanks very much, Carol. Ladies and gentlemen, first of all, I'd like to pay my respects to the local custodians of the land on which we meet, and indeed all lands from which we come. Without their 65,000 years of toil and enjoying life, we wouldn't be able to thrive as we do today. This was a little bit of a challenge when I was asked to give this talk. I must say my parents lived in Toowoomba during their retirement for several years, so I sort of feel like a partial justification for me standing up here, even though I'm almost a sandgroper now. I moved over west when I was 14 and have visited here and other places in southeast Queensland, northern New South Wales since then. In case I run out of time, here's the summary. So you're perfectly welcome to have a cup of coffee after this, or do whatever you like. I want to give just a tiny bit of background on natural selection taxonomy chromosomes, and then talk about my PhD project and the instruction of my supervisor of go where the evidence takes you. And I think as a scientist that's been a lifelong guide for me. I'll talk about kangaroo paw genus, which I did my PhD on, and a place called Gingin Cemetery. There is a Gingin in WA. In 1977, I was appointed as the West Australian's first government conservation biology scientist, and a flora conservation, that is, and also policy officer. I'll talk a little bit about the major tasks of that job. I'll mention the groups of organisms and habitats that I've focused on. Throughout the talk, you'll notice that over the 50 years, we've had tremendous advances in ways of studying and managing plants. In work on pollination, DNA has advanced from allozymes into whole genome sequencing. GPS, we can all travel around now and press a button and know exactly where we are. And the IT databases that we have available are just phenomenal. We also have tremendous ex situ seed banking capabilities now that weren't dreamed of in my time when I first started. And restoration, and lastly, I want to just very briefly mention that as scientists, we need to start thinking about Australian ecosystems from a theoretical point of view, that is Australian, homegrown and focused on Australian ecosystems, not borrowing, as we did when I was taught at uni, and many of you undoubtedly, from the Northern Hemisphere. finally, I'll conclude just very briefly with a comment on mainstreaming with the community and Aboriginal involvement. So, as biologists, we all owe a lot to this gentleman for introducing the concept of evolution by natural selection. We should be living, eating and breathing that because it provides tremendous guidance in understanding nature. Little less known is that Charles Darwin in 1868, I think it was, wrote a long paper about whether primroses and cowslips were varieties or species. And this was the introduction of the biological species concept as distinct from a taxonomically based morphological concept. Darwin advocated that, yes, things have to differ morphologically, but we can do experiments in his day with breeding systems by experimental cross-pollination, raising hybrids, looking at their fertility, and use that information to help us decide whether things are species, reproductively isolated, if you like, or varieties. And that's been a guiding principle that I've found very useful again in this line of work. As things progressed before all these new technologies I just mentioned, people in this case, this is California, Harlan Lewis, worked on chromosomes. And that's almost a lost art. We have one of the masters here in the room, Dave Coates, but I'm looking forward to the time when we merge DNA approaches with chromosomal approaches to studying organisms, because I think we'll have profound new insights from that. This is just one example of two species of Clarkia at the foot of Yosemite National Park that are diploid. They differ by one chromosome and also differ morphologically. Before I started my job, the Native Flora Protection Act of 1915 prevailed in Western Australia. That was set up and ultimately administered by the Forest Department, and the amount of interest in the flora shown by the Forest Department is indicated by the colouring on this sign, where you can see it's the state's floral emblem, and they've got the reds and the greens slightly confused. So very little actual work was done by the Forest Department. I started my PhD with Dr. Sid James, the late Dr. Sid James, in 1974, and he was a student of Sydney's Spinney Smith-White, who some of you may have read about, if not heard of. And Sid worked on chromosome numbers in what was then called Lobeliaceae. He was one of several students Spinney got on to look at major Australian families to ascertain whether there was chromosomal variation. But Spinney's interpretation of that variation was based on an idea of relaxed selection as the immigrant flora from somewhere else moved into Australia. Sid happened to look down a microscope when he was passing through Western Australia, a town near rock near Merredin, where this thing that's now called lithostoma was isotoma in Sid's day, isotoma petraea. And he looked down the microscope, and instead of seeing 7 pairs of bivalents, which he'd seen from New South Wales all the way across the continent, there was a ring of 14, all joined up, as you can see in one of the diagrams here. That is a phenomenon known as complex heterozygosity, and Sid devoted his career, in fact, to studying this plant and that system, with interest in other groups as well. But he kept right up till the end working on this with PhD students. Rather than relaxed selection, he could not conceive of the assembly of this chromosomal system from 7 bivalents to four chromosomes, or 2 chromosomes joining up and ultimately all 14 in a ring, unless natural selection drove it. And he argued convincingly and compellingly against both the Northern Hemisphere interpretation of this system, was known in Oenothera in Sid's day, and one other genus, I think, and the interpretations offered by biologists were stabs in the dark and certainly not driven by a concept of natural selection. Spinney Smith-White was a generous supervisor and said, "Sid, go where the evidence takes you," which Sid did, and I still remember him saying the same to me and my fellow students when we worked with him. Oh, there he is there. Sorry, I'm looking at the wrong screen, aren't I? So you're probably scratching your head as to what I was talking about when I said the ring of... Anyway, you can see a little ring just down there. Okay, I'll look at the right screen from now on. Gingin Cemetery was a student project when I was in doing second year botany, my third year at uni. I took 4 years to get my undergraduate degree for various reasons. And it was a project that had 2 species of Anagasanthus, the red and green kangaroo paw and cat's paw, and a few hybrids between them, about 40 hybrids, in a place that was annually slashed and burnt. And Gingin taught me several things, and I still continue to study the place. In fact, we've just got a paper we're working up now comparing this with Darwin's orchid bank in England, because they're classic comparisons of old versus young landscapes. So the sort of lessons that we learned was hybridisation was rare in this situation. There were 10,000 red and green kangaroo paws in the population and about 2,000 cat's paws, and as I say, 40 odd hybrids. And this is where I was inspired to undertake research, because I asked the lecturers at the time, has anyone worked on kangaroo paws for a PhD? And no one had. We had basic taxonomy done, and that was it. There was unexpectedly rapid turnover of whole plant communities over the 1.5 hectares of this cemetery, from those on dry sands to slightly moister sands lower down the slope. And the studies that I and my colleagues conducted demonstrated exceptionally limited seed dispersal and pollen dispersal, even though red wattlebirds were the major pollinators. We followed their individual movements meticulously and found that the overwhelming majority of movements were over 1 or 2 metres. We also discovered, when they ran a bobcat to clear land for more graves, that removing topsoil in this community just destroyed it. Overnight, after decades of slashing and burning and surviving as a native plant community, herbaceous community predominantly, the bulldozer just converted the place into a weed city. So veldgrass and the like just took over. And there were unexpectedly complex biological relationships. For example, poisoning kangaroo paw seeds with a cyanide derivative makes them germinate. So get your head around that one. So my PhD thesis was done and finished and awarded by 1980. The job came up because in 1977, flora amendments were made to the Wildlife Conservation Act, and this transferred the responsibility for flora conservation to the then Department of Fisheries and Wildlife from the Forest Department. And the director of the day, Bernard Bowener, convincingly argued for 6 new positions, one of which was, he thought, going to be a policy officer, and he was convinced that research should be part of that. And in these amendments, there was a section on rare or endangered species of flora, what became known as the declared rare flora. That was the first endangered species legislation for plants in the nation. Now, legislation is fundamental, and here's just a few acts that have influenced my career in various ways. So the Wildlife Conservation Act I've mentioned. Conservation and Land Management Act created a merger of Forest Department, Fisheries and Wildlife and National Parks, and has delivered us right through to the present day under 6 names, the present Biodiversity, Conservation and Attractions Department. The last word is the one that you focus on in that department. The second word is one you secondarily look at, and the first word, called biodiversity, is often not given much priority at all. The Commonwealth Environment Protection and Biodiversity Act you're all familiar with. Another act that was relevant to Western Australia was CITES, the Convention on Trade in Endangered Species, because early in its declaration it was decided to list about 1,500 species of Western Australian plants known to be either used as cut flower or seed in the wildflower trade. But the important point of all these acts is they rely on definitions of plants, species and communities. We have to understand them. So the challenges that I faced in 1977 were to identify what classes of flora should be protected, and that ended up being vascular plants, and fern allies, and flowering plants, cycads, conifers and the like. Declared rare flora, we needed a system to how do you declare rare flora, what evidence do you need. We also had to define presumed extinct plants, and devise permits to take flora for scientific research or commercial purposes, and also manage the nursery and commercial wildflower industries. So I spent the first 10 years of my life, much to my wife's aggrievement, when we had young kids, about a third to a quarter of the year I was out bush looking for native plants with colleagues. There was a preliminary list based on herbarium specimens that Neville Marchant and Greg Curie published in 1979, that green-covered book, and we figured that we needed to get out on the ground and ascertain whether things were genuinely rare or just poorly collected. In the process, we also were working on EPA directions to recommend new reserves, national parks and nature reserves. Now, the exciting thing about the West Australian flora, and probably the flora here and other parts of Australia, is that the discovery of new species continues in an escalating way in our time. Western Australia, the flora was regarded, Southwest Australia, as having 3,600 species by Joseph Hooker in the middle 1800s, and that hadn't changed by the time I started as a PhD student. Everyone thought the work was done. No new taxa of note really likely to be found. This graph just shows you that it was then and remains a complete fallacy. In my time over the 50 years, the estimate of the size of the vascular flora has doubled for the state, and the same for the southwest. There's also, therefore, it became very clear that there's this inexhaustible number of species needing attention and conservation assessment. It simply wasn't possible to do as a government agency on your own. So community involvement became essential, and we trialled the Banksia Atlas as a first citizen science mapping program, a national program, and that really helped us enormously, discovered 2 new species, and enabled accurate assessments of what things genuinely needed conservation attention. The Wildflower Society, likewise, has now run a program for 30 years under the leadership of Greg and Bron Curie, and written up with the help of Neil Gibson, looking at quadrat-based assessments of flora across parts of the southwest. These are the organisms that I decided to work on, because no single individual can do anything remotely trying to tackle 12 to 13,000 species in the whole state. But I decided to continue with the Hemideraceae, the kangaroo paw family, and there's the common cat's paw, the most common species, the orange thing, and one of the rarest subspecies, Anaxanthus bicolor minor. I worked on eucalypts and also orchids as well, and decided I should also take a look at a community that had been largely ignored when I first started, and that was granite outcrops. Now here's a series of books just to illustrate the progress in understanding Inselbergs or granite outcrops in Western Australia. The same applies worldwide. In fact, in 1972 I did a third year field camp and saw my first granite outcrop. We almost died from cold exposure too. We weren't forewarned that it gets pretty bloody cold out at Hyden. But the first conference on granite outcrops published internationally, multidisciplinary conference, was done by the Royal Society published in 1997, and other books have appeared, and that series of publications continues right up to the present day. So all of us know about this. The literature today is tremendously richer than it was 50 years ago. What we did discover after 10 years of surveying for plants to ascertain their genuine conservation status was that there was a correlation with those well-surveyed threatened plants and landscape position. And that correlation was roughly 2/3 occurred on old infertile uplands. So I'm talking about granite outcrops, lateritic mesas, Stirling Range and high sandplan in the wheat belt. 2/3. So if you wanted to look for threatened plants in WA, the first practical suggestion is go up. And you don't have to go up more than 100 metres quite often. So what we call a mountain in WA is not quite what you call a mountain over here. But that's where they were really concentrated. The lowlands around wetlands and the coastline, only 10%, 11% of the threatened plants were found there, and then in between about a quarter. So you'd think that's pretty compelling stuff, and you would think that the Department of Biodiversity would take heed of that and alter its management of uplands, basically, because of the concentration of threatened species that are there. When CALM was first formed, I remember talking to one of the fireys who was running the aerial ignition program, and he said, "Oh yeah, granite rocks, we use them for target practice." And I explained to him the significance of them, and slowly things changed and they stopped using them for target practice, except now they're using them for target practice. So if you stick around long enough, new people come in, new ideas, that sort of thing. Anyway, a little bit more of that later on. I just want to mention briefly ochbills, because I've got an opportunity tomorrow to talk a little bit more in more detail about them. But these are old, climatically buffered and in fertile landscapes. They're abundant in southwestern Australia, and they embrace these uplands that I've been talking about. And by old, I mean really old. So as a landscape, present for tens of millions of years. Climatically buffered, likewise. Ocean on two sides of southwestern Australia for 90 million years or more. And infertile in the sense of being phosphorus-limited soils, not nitrogen-limited soils. And as it turned out, these things are everywhere. And I'll talk about that tomorrow. By everywhere, I mean across the globe. So where were the declared rare flora populations by 1990? About a quarter were on land managed by CALM, so National Parks, Nature Reserves, Forests. 22% on road reserves. So that's the second most important category of land, and 16% on private land. And you can see the other statistics just there. So we had to do something special about road reserves. And one of my earliest tasks was to sit down with Main Roads engineers and say, well, how can we care for road reserves through the operations of your staff? And came up with the hockey stick, the inverted hockey stick, the yellow post, which has worked a treat. So the grader drivers don't need to know anything other than when you see one of those, you stop bulldozing, and when you see another one down the other end, you can start bulldozing again. And that's protected hundreds of populations of threatened species on road reserves. Sharing views across Australia was very important, and in my day, I participated on the CONCOM working group on endangered flora from 1981 to 1988. Those days, ministers appointed people to do that sort of stuff. And I also, when I moved to Kings Park in 1991, then participated on what's affectionately known as Charbag, the heads of Australian Botanic Gardens. And of course, APCC has now become the central organisation that provides these fora for sharing ideas across the nation. In listing a threatened flora, it became very apparent to us with this tremendous exponential increase in taxa being discovered that there were many things on the indeterminate list that we simply hadn't got around to looking at all. And with Mark Bergman, who worked as a consultant for me at that stage, we came up with this system of what's called priority flora. So four different categories, starting with priority 1 is known from less than 5 populations on non-conservation land. So under greatest threat, if you like, because of land status. And priority 4 is well surveyed and just keep a bit of a watchful eye on it. The black bars that you can see highlighted with the red arrows there are presumed extinct. And you'll see in southwestern Australia it's not going in the expected direction at all. And this was just evident that we knew so little about the flora that things that were presumed extinct weren't at all. They just hadn't been seen in the last 50 years. So the number has been declining since these records have been kept. Here's just one example. The only eucalypt presumed extinct in Australia, Eucalyptus rameliana. Lots of expeditions out to where Ernst Giles collected it. He said, gave the informative locality of beyond the Alfred and Murray Range. So Alfred and Murray Range is about 300 kilometres west of the Northern Territory border in the Gibson Desert. It turned out it was 500 kilometres beyond the Alfred and Murray Range. So several expeditions trying to find it where Alfred Murray Range, and it turned out it to be in the little sandy desert, not the Gibson Desert. So there are no, as far as I'm aware, no eucalypts, even though the genus is so large, that are presumed extinct in the nation. Some are getting pretty close, but there's none that are presumed extinct, and that's an extraordinary opportunity. Threatening processes you know all about. When this slide was done, 5 minutes? Yep. 2,500 taxa, roughly, we had to deal with in Western Australia when the total flora was estimated at that time at 8,500. Root rot disease, weeds, usual story. Dave Coates kindly lent me this slide a while ago, and it just highlights the range and extent of different threatening processes that apply to the flora. So what do you do about it? Kingsley Dixon came up with this diagram for an integrated approach that starts with alpha taxonomy, applies conservation genetics, and then propagation science, germplasm conservation, restoration ecology, and ultimately achieves recovery. Recovery teams are all the go. 20 years ago now, I guess. And lots of recovery plans have been written, interim recovery plans, if not full recovery plans. But very little research and experimental management in Western Australia was actually funded based on the recommendations of those plans. We went into regional management programs for threatened flora, and that proved very useful. So combining all the threats in a specific region. A WA Threatened Species and Communities unit was established. Seed centres were established, and Kings Park became involved as well. So this is almost a penultimate slide, I think. To summarise what's been done about it, governments and ministers basically over the 50 years I suggest have changed their roles from being chairman of the board to be chairman and chief executives, and that's been to the detriment of conserving for threatened flora, in my view. Statutory authorities and government departments have had to change their philosophy in Western Australia, certainly, and have become meek and mild and don't upset the minister as the operating guidance, not care for biodiversity. Mainstream media, the West Australian used to have a weekly supplement called Earth 2000 that was 8 to 12 pages long when I first started. Now the only environment stories they print are available from the Northern Hemisphere usually. University research and teaching. I tried early on to get university people interested in flora conservation and failed miserably, even though I had money. But a lot of work now by industry and consultants, by community organisations, private individuals and Aboriginal people. I won't dwell on that slide. I've got two more, Madam Chair, before I finish. This is something I really wanted to emphasise because when I was a scientist and trained, we were taught publish in the scientific literature, job done. And nothing could be further from the truth in our game. If you look at the percentage of, this is British data, but look at the percentage of reasons that people apply to pick up new knowledge in Britain on conservation, and you see that primary scientific literature right down the bottom, only 2.4% looked at it. So you've got to communicate what you find out is take-home message. And lastly, we really have to walk together with Aboriginal people if we're going to do a proper job. We've been finding out tremendous things in Noongar country, and I'll talk a little bit about that on Tuesday. Thank you. Yep.
Okay, thank you very much, Steve. From my perspective, your work has influenced not only my research but my teaching, and having examples from the Australian flora is just critical. Even though when I started out, looking at the Northern Hemisphere flora was what we did.
Yep.
So thank you for your lifetime of work in this area. Also resonating for me was that last slide on Sutherland's work from the UK about how our work has to be communicated for people, for practitioners to use that. And I think we've all missed that for a long time.
Absolutely.
And some of it's because we don't get to conferences and so forth and do that. But so now we have the opportunity to ask you, Steve, some further questions. We've got a bit of time here. Some questions from the audience. If you could just say your name and where you're from, thanks.
Bob Makinson from Sydney. Steve, the West Australian system of identifying priority flora and having a list of sub-threatened species, if you like, and actually taking a systematic approach to finding out more about those is pretty much unique in Australia. I don't think any other state is doing that. Instead, the other states, including my own of New South Wales, sort of freak out about the number of threatened species that are suddenly arriving on the list, and not having looked at them, they know nothing about them when they get there, and therefore no conservation action. How do you think we generalise the lessons from that WA system of prioritisation and investment in flora before they hit the threatened list?
That's a great question, Bob. I really can't answer it other than to say every jurisdiction is different. We fortunately had a department and a department head who believed firmly in a scientific evidence-based approach to conservation early in the day, and it took a long time for the government to work out that that's getting in the way of development. 50 years on, that's now happening in a big way. So, you know, even the EPA, which was established in 1971 or 72 in Western Australia, that act is being revised so that the EPA no longer has to look at the environmental concerns that were its primary focus, but also social and economic. So they're no different from Cabinet, basically. So, you know, it's not entirely rosy in the West, you know, and I guess the take-home message is that things can change without eternal vigilance from the broader community. Things can change and go bad or good depending on how the pollies react. So all I can say is our system has worked reasonably well. Dave took over when I left the department, and, you know, it still was working. We were just talking now about how bureaucratic it is to try and get anything listed in Western Australia. So maybe it's going to get tougher in the West as well, but at least it's a model that's been out there and been in practice for several decades. So it's a model, and it's up to other jurisdictions to act on it as they can and wish.
Thank you. Okay, one more question. Over here.
My name's Jan Aldenhoven, and I live on Minjerribah, North Stradbroke Island. We also have some rocky outcrops there.
Yep.
And I was just wondering why you think the diversity is higher, and is that trend applicable to different types of rocks? So in our case it's rhyolite, and there is a very high diversity. And which minerals is it related to?
That's a really good question. What you live on and the rocks you describe is another. The Ocbill Yodful division was a simple binary that we set up early on. It's actually a cube, and another point on the cube are called Ocfells. So a lot hinges on the bedrock. If the bedrock is rich in minerals, then the soil will be fertile, not infertile. So hence Ocfells, old climatically buffered, fertile landscapes. They've been around the same period of time as Ocbills quite often, tens of millions of years, they can be. Climatically buffered, that depends where the oceans have been, adjacent to your coastline. And they can be equally as rich in endemics, we found. Western Australia doesn't have too many of these fertile rocks in the southwest. We have dolerite intrusions in granite, but hardly big ones. And so it's not a big deal, but it certainly is in South Africa and parts of Brazil. So it's perfectly likely that if your outcrops have been old landscapes, literally up above sea level for tens of millions of years, or millions of years, shall I say, that the body of theory that applies to the infertile landscapes would also apply in your situation. I've also had someone challenge me on rainforests up and down the east coast for the same reason, because they're usually on more fertile soils than we're used to. So places like Hawkesbury sandstone, classic Ocbills. You've got them all through Queensland here, the flat top mesa sort of inland stuff. But rainforests on fertile soils, that's due to geology, and you can still have old landscapes that have fertile landscapes. Well, those three reasons are the hypothesis, old, so that's testable by geological investigation and molecular phylogenetics. Climatically buffered, the same. You can look at where oceans have been in the past. There are good models for that worldwide. And the fertility of the soil you can test, so you can work out whether it's rich in phosphorus or rich in nitrogen.
Those 3 things combined cause speciation.
That's right. Yeah, and if you want to bore yourself, you can get into read some of the papers, and you'll find the logic of the argument is presented in more detail than I can here and now.
Or lack of extinction.
Or lack of, well, that's quite right. Dave just said, or lack of extinction, which is another feature of Ocbills that we're finding. They have depressed extinction rates, not heightened speciation rates.
Okay, can you move on? Thank you very, very much. Okay,